Good morning, students. Today we will be discussing about the histology of kidney. So you can see I put the actual photo micrograph of the kidney. As all of you know, the kidney consists an outer part known as the cortex and inner part as the medulla. So this is the cortex and this is the medulla. So inner part is the medulla, the outer part is the cortex. So what are you seeing in the cortex? The most important identifying feature of the kidney is the presence of renal corpuscle. Now this is one renal corpuscle, this is one renal corpuscle, you have one more here, you can see one more renal corpuscle. Plenty of renal corpuscles are present in the cortex. Now what are these renal corpuscle? The renal corpuscle is nothing but Bowman's capsule. In the middle you have the tuft of glomerular capillaries. So Bowman's capsule with the tuft of glomerular capillaries in the middle or the center is known as renal corpuscles. So this is the most important identifying feature of the kidney, especially the cortex. So once you see the renal corpuscles in the cortex, you can say that this is a slide of kidney and it is especially the region that is the cortical region, that is the cortex. Now what are the other things you have to observe here? You have to observe the proximal convoluted tubule and distal convoluted tubule. Now look at the proximal convoluted tubule. It is circular of course, but the lumen is cloudy because it is filled up with microvilli. So all the cells of the proximal convoluted tubule have brush border appearance with plenty of microvilli, which makes the lumen cloudy. And one more important thing you see is that the PCT is darkly stained. So the proximal convoluted tubule or the PCT is darkly stained. The cells have round nucleus. Lumen is cloudy, not very well clearly seen because it is covered by the brush border or the microvilli coming out from the apical surface of these cells. Now look at the DCT, the distal convoluted tubule. The lumen is quite clear, quite large, it is clear, and the cells are not as darkly stained as the PCT. So the DCT or the distal convoluted tubule, it is not darkly stained like the PCT. So this is the proximal convoluted tubule, this is the distal convoluted tubule. So what are the important identifying points in the kidney? First is the presence of renal corpuscle, that is the Bowman's capsule with the tuft of glomerular capillaries in the middle. Then you see the PCT, dark staining PCT, light staining DCT. Apart from this, we will see in the medulla there is no renal corpuscle. So this is the kidney and it is the medulla part. So the inner part of the kidney is known as medulla. So here you don't see the renal corpuscles, but you see plenty of tubules. What are these? These are the loop of Henle. These are the loop of Henle. So the absence of renal corpuscles tells us that this is actually the kidney and the medulla part. So in the kidney, the medulla does not have renal corpuscles. Instead, it has plenty of loop of Henle. So the cortical region, you can see the cortical region and the medullary region. The medullary region. Thank you.